Hello, and welcome to the Fascinated by Fungi podcast with me, Dr. Gordon Walker. Today we're going to go over the broad array of different mycorrhizal relationships. So those are fungi that are associated with the roots of plants. And we'll talk about some of the different kinds and the way that they interact and how they're different. And I'll touch on the concept of the wood wide web, or the idea of a sort of mycelium as the nature's internet. The more sciencey term for it is the common mycorrhizal network. And I'll go over some of the sort of competing theories about, what's going on there and how much we really understand versus what we're still working to understand experimentally and based on the data that we have. So I want to start out by thanking my sponsor, North Spore, a phenomenal mushroom company that's up in Portland, Maine. And because it's getting cool, because it's fall, and because winter is just around the corner, it's a great time to grow some mushrooms that like cool temperatures. So I just got a new box worth of stuff from North Spore. I got some snow oyster mushrooms, as well as piapini. I've also grown namicos and king trumpet mushrooms this time of year in the fall, going into winter, and have had great success with the kits from North Spore because those mushrooms like cool temperatures, and I think they're really delicious. Snow oysters are super meaty. Piapini or piapinos are sort of velvety and beautiful brown mushrooms with a lot of good sort of earthy flavor, but not overwhelming like the portobello. And namicos are very slimy, but they're super good in soup, and I like them in stir fries. King trumpets are just big, meaty mushrooms. So those are all ones I highly recommend, and you can get affordable, good kits from North Spore. They will always fruit out, and they've got great recommendations on special things you can do with each variety to ensure successful fruiting. So I also, this time of year, like to take the North Spore Immunity Blend. This is pretty much the only mushroom supplement I take on a regular basis. And I like the North Spore supplements because they're made with whole fruiting bodies, which pound for pound have more bang for your buck than mycelium supplements do. I have data to show that and have experienced that personally. So I prefer to take mushroom supplements made with whole fruiting bodies, especially because the main component you're going to get out of a mushroom supplement is the polysaccharides, and those are what have actually been demonstrated to have medical benefit to our system as immunomodulators. So from a scientific perspective, taking whole fruiting body mushroom supplements is going to be more useful for your immune system than just taking mycelium or something else that is like tincture or liquid extraction based. So that being said, thanks to North Spore for sponsoring this episode, and let's dive into mycorrhizal relationships and what they are, and where they happen, and all the things about them. So mycorrhizal networks have been often referred to in popular media as like the wood wide web or the internet of nature. And this is a great gateway concept to thinking about what's known in science as the common mycelial network. It's the idea that underground different plants are potentially linked by connections of mycelium to each other, and that there may be transfer of nutrients across that mycelium, as well as communication molecules, and that all the trees are working to sort of help and buffer each other within a forest ecosystem. And so this has been proposed, and like I said, it's a great gateway concept. But within media, it's very popular to sort of depict, this broad-ranging network of mycelium under the soil, linking all these plants together in this sort of big mutualistic community. And there's been a lot of questions, especially recently, about like how true is that assumption, and how much of it is based on our sort of anthropomorphic assumptions about what is actually happening versus what we can measure experimentally. So I'm going to spend the first part of the podcast going over the main types of mycorrhizal relationships, differentiating them, and then at the end I'll come back a little bit to some of the controversy and ideas where people have been a little skeptical of this whole wood wide web concept. And I think there's value in it, but there may also be a chance that we've overstepped what we actually know and are just sort of assuming that things work the way that we want them to without actually paying attention to what the data says. So let's dive into what mycorrhizae are. So mycorrhizae are fungi. It's the mycelium of fungi that are associated with the roots of plants. So mycelium is sort of the thin network of hair-like threads composed of hyphae, which are sort of tubular long fungal cells. And mycelium is present in most kinds of fungi, especially terrestrial fungi. There's saprotrophic fungi, which grow their mycelium into like soil and wood and decompose stuff. And then there are mycorrhizal fungi, which the mycelium of which is associated specifically with the roots of plants. And they are different lineages of mycorrhizal fungi. Some evolved on their own, where some other ones evolved from saprotrophs. And so they have different capabilities depending on what their genetic history was. Mycorrhizae are the fungi that, again, have co-evolved with plants. And they've been present for at least the last 400 million years. And very much the evolution of plants was driven by fungi and vice versa, in that they probably came to land together. And plants really only able to survive on land because of fungi. Fungi probably would have been able to survive on land to some degree without plants. Lichens are able to exist in very extreme habitats, but they're still dependent on a photosynthetic partner, usually an algae or cyanobacteria. But it is the sort of co-evolution of plants and fungi together through the building of these mycorrhizal relationships, which probably started out originally as sort of like pathogenic parasitic type relationships, and over time the antagonism of fungi and plants together created this like mutualistic symbiosis. However, that symbiosis is also somewhat dependent on a little bit of a give and go, what they call like a marketplace or economy of nutrients between plants and fungi. So the relationships are conditional, very much dependent on what's going on in their environment and what other organisms are at play and what kind of nutrients are available, and temperature and moisture and all this kind of stuff. And even the benefits that fungi confer to their hosts can vary. Sometimes it's a big benefit, sometimes it's sort of a medium benefit, sometimes it's a little teensy benefit, and sometimes mycorrhizal fungi can actually hurt their host under certain types of conditions. So again, adding a little bit of nuance to what we think about when we mention mycorrhizal relationships is that it's not always 100% positive. Most of the time it is, but again, we're learning about more of these exceptions and trying to better understand the spectrum of these fungal plant interactions. So, there are, so mycorrhizae are a really important part of what's called the rhizosphere. So basically the whole sort of microbiome of the roots of a plant. So the fungi or the mycorrhizae are really important of that part of that rhizosphere, but there's also, other kinds of soil fungi. There are lots of bacteria, there's small swimmy protist things and animals. So there's a lot of different organisms going on in the rhizosphere, and mycorrhizae often help to sort of sort out, the pecking order or increase the diversity of the bacteria, and usually they exert positive effects on the rhizosphere. But it's more than just the plant and the fungi down there. There's a lot of other complicating factors that, again, make this a nuanced thing, that it's not just the internet of nature, right? So the role of mycorrhizae in soil is typically to help absorb micronutrients and water. That is the main benefit that the fungi is giving to the plant. And in exchange, the plant will give the fungi sugars and sometimes lipids to help mycelium grow more. And then it's a symbiotic, mutualistic relationship. Mycorrhizae also has the capacity to stabilize soils. So by creating lots of little hair-like threads, it can cause little micro aggregates and keep soil in place. Mycelium is really good at absorbing water, so it also increases the water holding capacity of soil. It will modify the pH, usually bringing it down, since fungi exert or excrete a lot of protons. It will usually increase the microbial soil diversity, which is generally a good thing, or at least it will help to sort out the microbial soil diversity in a way that is beneficial for both the fungi and the plant, again, usually. It can help with mineral liberation or making nutrients more bioavailable within the soil. And it can also help to sort of bind up heavy metals and help remediate toxins in the environment. So all of these things that mycorrhizae do are very helpful for plants that are trying to grow in soils that might not necessarily be ideal. But there are different types of mycorrhizae for different types of soil as well, and that's what I'm going to dive into now. So a lot of what I'm going to mention here comes from a paper by Hawkins et al. Mycorrhizal mycelium as global carbon pools. It's a paper from Toby Keir's lab, and it's a really good breakdown of the sort of global carbon flux and the impacts that these different types of mycelium have on this global scale. And they reviewed a lot of different studies to kind of create these numbers. But within their paper, they also put out the fact that it's not 100%. This is stuff that we're still guessing at. We don't know. So I'm going to tell you these numbers with the caveat that as we learn more, they may change. And there's potential that things could be different, although the trends that they've laid out are probably pretty true. So global impacts of mycorrhizal networks. Right. So first, I just want to bring up there's two main types of mycorrhizal relationships. There are what are called endomycorrhizal fungi, and those are fungi that create structures within the plant cell called arbuscules. And there's ectomycorrhizal fungi that create sort of a sheath around the host root, but don't actually penetrate the plant cell. So there's really only one kind of ectomycorrhizal fungi. They're formed by both ascomycetes and basidiomycetes, and they're a little bit different depending on whether it's an asco or basidio, but it's generally the same sort of sheath around the root as far as I can figure out. And that's very rare comparatively. It's only associated with about 2% of plants. So 90% of plants have a mycorrhizal partner, but only about 2% of those are ectomycorrhizal. Then there are what are called arbuscular mycorrhizae, which are a kind of endomycorrhizal association, and that accounts for about 72% of mycorrhizal associations with plants. So ectomycorrhizal associations are important because those are the ones that form mushrooms, but arbuscular mycorrhizae are formed with glomeromycota. They don't form mushrooms, but they're far more common and widespread. And then there's a few other weird ones: ericoid, orchid, arbutoid. I'll go into a couple of those other alternative types of mycorrhizae later on. But right now I really just want to focus on the difference between ectomycorrhizal and arbuscular. So it's sort of what I'm going to call ECM is ectomycorrhizal and AMF is arbuscular. So keep those definitions in mind as I talk through this. So from the paper, the Hawkins et al. Paper, there's about over 40 million years of association with plants that mycorrhizal fungi have. And about 75% of the terrestrial carbon is stored underground, potentially in mycelium, which is a pretty major amount of the carbon that's in the soil. So in general, plants tend to give about 3 to 13, or sometimes quoted as like 4 to 20% of the carbon, the sugar that they make, down to the mycorrhizal fungi on their roots. So that's a fairly significant amount of the carbon that they produce. The rest of it is going towards plant growth, but up to about 1/5 of the carbon they make can go down to the roots. And in some situations, I think there's a paper they cited where they said that it was up to like 50% of the plant's carbon went down to the roots. So that's also pretty major. And that's not typical, but that can happen. So total, and this is based on the massive sort of calculations they did, about 13.12 gigatons of CO2 equivalents are sunk into the soil every year through mycorrhizae. And that accounts for about 36% of the global fossil fuel emissions. So it's a pretty significant chunk of what human beings are pumping out into the world every year. And if you think about the more we cut down forests, we're losing, and this number of 13.12 gigatons is going down because we keep cutting down more trees and we keep burning more fossil fuels. Not a great combination. This is why global warming is happening. So Uh, of that 13 gigatons, about 3.93 gigatons of CO2 equivalents are sunk into the ground by arbuscular mycorrhizae, AMF, which are associated with about those are associated with about 57% of all the vegetation on the planet's surface, but aren't actually sinking that much carbon overall, even though they're associated with a much wider percentage of plants. So ECM fungi, the ectomycorrhizal ones, sink about 9.07 gigatons of carbon CO2 equivalents into the soil every year, and they are associated with only about 26% of the vegetation cover. So they are associated with less plants overall, and only about 2% of plants are actually associated with ECM fungi. So that 26% of veg is big boreal forests, so big tracts of conifers up in like the Arctic and stuff like that. And so there's a tremendous amount of carbon that is sunk into the soil by those, temperate and boreal forests near the Arctic and Canada, Siberia, stuff like that. But again, those are some of the forests that we're cutting down still, and so we're losing a lot of this carbon sinking capacity. And in a much smaller percentage of the total carbon flux, there's also about 0.12 gigatons of CO2 equivalents sunk by ericoid fungi, which are kind of mycorrhizal fungi that are associated specifically with like Ericaceae, so like blueberries and cranberries in these sort of wet bogs and heathlands, high elevation, wet, cold places. And so it's not a huge percentage of carbon, but it's significant. And then, also thinking about like with the different types of mycorrhizae, you have like internal and external mycorrhizae. So some of the mycorrhizal mass is actually inside of the plant root. It's not always external in the soil. It's both internal and external, but that's part of where that carbon is getting sunk. And I'm going to sort of talk about some of the differences here between AMF and ECM fungi and just help you understand, because this is one of those things that I didn't even know. I thought all mycorrhizae was the same. It's not. And it wasn't until I heard an interview with Tom Burns from Berkeley that I really came to understand the difference there between AMF fungi and ECM. And so I'm going to try to dive kind of as deep as I can, but please keep in mind that I don't have an official academic background in anything to do with mycorrhizae. I am a yeast biologist, and so this is my interpretation of reading as much as I could and trying to synthesize stuff over the last couple of days to be able to do a podcast and talk to you guys about this. So this stuff that I'm about to quote is coming mostly from Luzzoni et al. 2018, The Contemporaneous Radiations of Fungi and Plants Linked to Symbiosis. I'm trying to cite some of the papers that I was doing research on to do this episode. So, yeah. Sorry, I have a big page of notes here, and I'm just trying to like make sense of it all and make sure I'm not missing things as I go along, but we're getting there. So arbuscular mycorrhizae emerged something like 520 to 420 million years ago, and they came up with the advent of land plants. So they associate with non-vascular plants like mosses and liverworts and ferns and also early conifers. AMF fungi are a relatively small section genetically. There's something only like 1,000 of them in Glomeromycota. So there's not a lot of genetic diversity compared to like ECM fungi, where there's thousands, if not more, species. There's relatively few Glomeromycota, but they're very common and they're very widespread. And there may well be more species diversity there than we recognize, but it's sort of an undersampled thing because it's just in soil everywhere. I know every time that we take a cubic inch of soil from anywhere on the planet and do DNA sequencing on it, we discover new species. So again, some of these may be undescribed or understudied simply because Glomeromycota and AMF fungi do not form mushrooms. So you can't find the mushroom and then look for the mycelium. You just have to sample the soil. Glomeromycota do tend to have like very large spores, and sometimes you can even see those like big circular spores in soil, and they are obligate biotrophs, which means they only form in conjunction and live in conjunction with a living plant, whereas a lot of ECM fungi evolved from saprotrophic fungi, and so they can exist as like mycelium separate from the host plant a lot of the time. So they're more facultative. So the ECM fungi emerged something like 400 to 250 million years ago. And again, there are both ascomycetes and basidiomycetes that form ectomycorrhizal relationships. So that's part of why there's such a large range in between like when they evolved. And generally those came up with conifers, and when there started being a lot of gymnosperms, that's when these ectomycorrhizal relationships evolved and helped probably conifers conquer a lot of the planet, become one of the predominant major plant species. And even still, like the oldest, most mature forests tend to be conifer dominated and have a lot of ectomycorrhizal relationships. So angiosperms came up something like 120 million years ago, and angiosperms are interesting. Those are flowering vascular plants, and they can integrate both AMF and ECM fungi, and that's been hypothesized to be part of why they're so widespread and flowering plants are so successful, because they were able to kind of integrate both types of ectomycorrhizal relationships. And when we think about like species succession, we talk about going from like a grassland to like a fully mature forest after a disturbance, say a wildfire or human disturbance, us cutting stuff down. It generally starts out that in bare soil or kind of a grasslands, you have nitrogen fixing plants, so legumes. Those are stuff like peas, garbanzo beans, that don't necessarily have mycorrhizal relationships. Instead, they're dependent on bacteria. So they set up a little node on their root where they invite bacteria in, and they shift around the conditions and then cause the bacteria to start fixing atmospheric nitrogen into bioavailable ammonium, NH4. And that's important because plants can't produce their own nitrogen, but bacteria can. So plants are working in conjunction with bacteria to fix that nitrogen and help them in these sort of poor soils. There's also some fungi that can help to create conditions that then help nitrogen fixing bacteria do their thing in the soil. So fungi can also sort of cultivate bacteria in a way that helps them gain access to the nitrogen the bacteria are fixing from the air. So bare soil is usually starting up those legumes and some saprotrophic fungi and maybe a little bit of arbuscular mycorrhizae there as well in these grasslands and stuff like that. But at that point, the fungal interactions aren't necessarily that helpful. Once you get into sort of a middle-aged succession of stuff, so you start getting deciduous trees, broadleaf trees, hardwood trees, mixed forests, then you get a lot more arbuscular mycorrhizae and bacteria contributing to the sort of rhizosphere. And then later on, in a very mature forest, like an old growth forest, you'll have majority conifers with mostly ectomycorrhizal fungal associations. And that's where the plants are deriving the most positive benefits. But there are differences here between what AMF and ECM fungi do. So in terms of where they're found in the world, the AMF arbuscular mycorrhizal fungi tend to be concentrated around the equator in a sort of hot and wet environments. They're very common in tropical environments. They're common in grasslands. They're really common in like human landscapes and agriculture, places where we are farming stuff. And so AMF fungi are really important and highly studied in the context of agriculture, whereas ECM fungi tend to be mostly concentrated around high latitude places and also sometimes high altitude. And they prefer sort of cool and dry habitats and these like boreal and temperate forests that tend to be hardwood and conifer dominated particularly. And with this is also, so that's ecological succession I talked about, and then also, you know, the sort of latitudes across the planet. But along with latitude, there's altitude. So in addition to altitude, you know, lower things tend to be grasslands, and then as you go up in altitude, you start to hit temperate forests. So grasslands are mostly arbuscular mycorrhizal fungi with high amounts of available nitrogen. Temperate forests are more ECM dominated fungi, but there tends to be AMF fungi with like the plants on the understory, so all the little plants growing on the ground. In boreal forests, again, as you go up in latitude or in elevation, it'll be mostly ectomycorrhizal ECM fungi with a little bit of ericoid, so it's like blueberries, cranberries kind of stuff as the understory. And those have a little bit more decay capability, so they're able to like generate some of their own nitrogen. And then at the highest elevations, you have heathlands or these like peat bogs, which are very nutrient poor. It's cool and wet and damp. And those are dominated by the ericoid mycelium or mycorrhizae with a little bit of ECM fungi. And those tend to have a lot of decay capabilities because they need to like generate their own nitrogen and energy. So here's a big breakdown of AMF versus ECM fungi, and I hope I got most of this right. There's a lot of information out there on this, and it's dense. There's a lot to read. There's a lot to understand. One of the papers that I got a lot out of because it had a great graphical abstract was dominant mycorrhizal associations determine soil nitrogen availability in subtropical forests by Lee et al. 2022. So this is where I got a lot of my information from, and just thinking about lots of different things. So arbuscular mycorrhizae fungi, AMF, have relatively little species diversity, as I talked about, some like 1,000, 1,200 species that have been sort of recognized. They tend to associate more with deciduous broadleaf trees and grasses and sort of non-conifers, although they do associate with some conifers. They're associated with about 72% of plants on the planet, but they do not form mushrooms. Instead, they're these big circular spores. They are obligate biotrophs, meaning that they only live with their plant hosts. They tend to form shorter networks. They don't form like long-ranging networks, as far as I know, because the cells on Glomeromycota, the hyphae, don't have septa. So there's not little like pores between them to help with nutrient transfer. So it tends to be just like one long cell as opposed to like many long cells in parallel that then transfer nutrients between them. And AMF fungi like inorganic nutrients and prefer really rich soils. So it's a low ratio of carbon and nitrogen, basically lots of nitrogen, and they can handle that. They're happy with that. They let like warm, wet conditions and higher pH or basic soils. They're really good at phosphorus uptake and have a lot of like phosphatase enzyme activity. They're lower at like nitrogen uptake, and they don't have a lot of nitrogen uptake enzymes, but they're sort of scavenging the nitrogen that's in their environment. They do tend to have like fast nutrient cycling, quick turnover of ammonia into ammonium, and then that can lead to more leaching. So if you're in a heavily fertilized environment, because they don't take up a lot of nitrogen, there's more leaching of nitrogen. They have a high mineralization capacity, so they're good at, I guess, keeping soils in place is one of the things that mineralization is doing. They're associated with high plant species diversity. So AMF have relatively low fungal diversity, but are associated with high plant species diversity. Again, in tropical, warm, wet environments, it tends to be a lot more plant species. Like I said, they form sort of shorter, localized networks. They have a faster generation time, so arbuscular mycorrhizae tends to grow and die and decompose faster, as far as I understand, than ECM fungi. They don't have as much water holding capacity because they don't form longer, wider, broader networks, and maybe something to do with the size of their hyphae and the cellular structure. They don't come from saprotrophic lineages, so I do believe there's some AMF fungi that display some ability to decompose the stuff that's around them, but it's not one of their main trophic modes. And they offer sort of less protection against pathogens than some ECM fungi do. But AMF fungi are very important, very widespread, and are by far the most common type of mycorrhizal associationship on this planet by a wide margin. And they've been around the longest as well. So ECM fungi, ectomycorrhizal fungi, do form mushrooms. Not all of them form mushrooms, keep in mind, but it's usually ascomycetes and basidiomycetes. And in theory, there's maybe some like Mycorrhizae stuff that can form something that's sort of ECM-like, but I'm going to just stick with saying it's ascos and basidios. It's only about 2% of associations on this planet are ECM versus like 70+% for arbuscular, so big difference there. But it's still associated with a fairly significant amount of the vegetation on the planet because big boreal forests and trees cover huge tracts of this planet, as well they should, because that's how they're sinking so much carbon into the soil, right? So the AMF fungi only sunk like 3 point something gigatons, whereas the ECM fungi sink like 9 point something gigatons of carbon into the soil every year. So they are responsible for sinking much more carbon into the soil, and thus, again, the importance of those boreal forests for our sort of global carbon cycle. And so ECM tend to be a little bit more associated with conifers, although they also associate with hardwoods and deciduous trees. They do form longer, wider reaching networks, and they have septa, and they're able to do more nutrient transfer within that mycelial network than AMF fungi, as far as I understand it. And they tend to prefer, they're facultative biotrophs, so they also are a biotroph, meaning they live with their host as opposed to like killing their host. But they're facultative because they can live on their own sometimes, and they what does it say? They're multiple modes, so they can also like do a lot of saprotrophy and decay stuff, as well as living in conjunction, that mycorrhizal association with their plant host. They tend to prefer poorer soils, so there's a higher carbon and nitrogen ratio, cooler, drier climates with low pH, acidic soils. They are lower amounts of phosphorus uptake, but higher amounts of nitrogen uptake. They tend to mine nitrogen, so there's even like foraging activity that some mycelium will like grow out along hyphae to go like seek for nitrogen in the soil. And this has been demonstrated with, I think, conifer forests up in the Pacific Northwest, where there's salmon die-offs, and then the trees that are near the edges of the streams and pools where the salmon die and rot are able to go to those. They're sending mycelium out to mine the nitrogen from that decomposing salmon bodies, and to me that's really cool, because it's demonstrating clearly that there's like different strategies. And also within this, for both AMF and EMF fungi or ECM fungi, there are different acquisition strategies. So some focus more on like eating what's right here in front of me versus like maybe there's something over there. There's a lot of different dynamics there that we don't fully understand, because it's really hard to study amorphous hair-like cells down in the soil, because you dig it up and immediately you change the environment, and there's not an easy way to take a cross-section of soil in any random spot and look to see how far a mycorrhizal network goes. And even, I know that like Merlin Sheldrake, the guy who wrote Entangled Life, did some really interesting like PhD work where he was in the forest literally like carefully extracting and digging up mycorrhizal networks to try to look how far they'd extend. And like that kind of thing is incredibly laborious, very difficult, and challenging from an experimental perspective. So we've only done this with a very limited number of species and trees, and so understanding the extent to which some of these networks go is problematic and still unknown. So ECM fungi have slower turnover of ammonia into ammonium, so there tends to be less leaching, but they're in low nitrogen environments anyhow, so their capacity to go mine and take in nitrogen is much more useful when there's limited nitrogen. They have a lower mineralization capacity, but I believe they're still really good at solidifying soils. They might even be slightly better at bioremediating heavy metals from the soil, because I know that a lot of ECM fungi will fix heavy metals in soil. They are associated with less plant species diversity, but more fungal species diversity, and that's partially in these like big boreal forests. It's pretty much all like one or two types of tree, but then there's a lot more fungal species abundance around them. So they cultivate communities where other fungi are able to grow. And keep in mind too, as I'm going through this, that one tree can have upwards of 10, 12, 24 or more fungal species associated with it, and there's probably a lot of angiosperms have both AMF and ECM fungi, and so they're all interacting down there in the soil, and there's going to be a pecking order to how they all decide what's happening and who's more useful or not. So I believe that ECM fungi tend to have higher water holding capacity because they form larger, wider networks, and they also have a longer generation time, so the mycelium lives longer than the AMF fungi in general, and so there's less turnover, and there may be more long-lasting connections there that ECM fungi form. And they have more capacity for saprotrophy or breaking down organic matter in the soil. And a lot of ECM fungi will break down organic nutrients to feed themselves, as well as getting carbon from their hosts. And that's probably part of why they're able to grow further and farther, because they have more capacity to kind of support themselves. So if they're further away from the host and the plant sugars aren't quite as near, they can start creating enzymes to break down organic matter in their environment, digest it, and then keep growing and seeking out more nutrients. And they're also apparently potentially give more protection against pathogens. So those are some of the differences there between AMF, EMF fungi. So in terms of like the establishment of these types of relationships, the pathway that arbuscular mycorrhizae use is sort of similar to the way that the nodules and bacteria legume relationship is formed. But the spore, the big circular spore, will produce fungal proteins, which then spur the production of signaling molecules from the plant. Specifically, these things called strigolactones produced by the plant that help to establish the connection there between the hyphae and the plant, and these Myc factors. And so generally, like the hyphae is growing out from the circular spore towards the plant root. It penetrates the tissue and then eventually penetrates inside of a plant cell and forms this arbuscule, which is kind of like a big knotted high surface area structure within the cell that is then doing exchange of both water and nutrients. And so the fungus is taking in sugars and lipids from the plant and giving back to the plant lots of water and phosphorus, nitrogen, zinc, potassium, and selenium are some of the main like nutrients that it's giving back. In terms of ECM fungi, they can exist both as a spore or I think also as mycelium, and make the decision then to colonize a root. And it's not as well understood how that relationship is formed. It's not dependent on these strigolactones in the same way, but it's guessed and hypothesized that it's a product of flavonoids produced by the plant root and possibly some hormones, but it's then forming a sheath around the host plant root. Instead of actually going inside of the cell, it's forming a sheath called the Hartig net, and that's very characteristic of what ECM fungi do versus AMF fungi. And so in conifers, that Hartig net penetrates pretty deep into the root. Again, it's not inside the cells, but it's still penetrating inside of the root in a meshment. And so it goes all the way down to the cortex of the root in conifers, and in angiosperms, it's a little bit more of a shallow association. But both seem to induce more root growth and really stimulate nutrient uptake. There's a sort of nutrient economy for this kind of exchange with these mycorrhizal associations, because again, like I said, like one tree can have like upwards of, 20 different fungal species on it. And some fungal species might be give you not very much phosphorus or nitrogen, but they might not require very much carbon either. So they'd be a very like low, if you had a baseball team, you'd have a couple guys who always sat on the bench, but you wanted to have them just in case, your star players got knocked out or something like that. So you might have a couple of these associations where it's like low effort, low return, but it's good to have a few of those hanging around. And then you might have other ones that are like your diva star player, maybe a Chantrell or Porcini. I'm going to assume those are diva star players. Why not? But those might have the capacity to uptake more nitrogen, more phosphorus, and be really beneficial for the plant host, but require a lot more carbon. Remember, some, it can vary from like 3 to 13% of the carbon a plant produces to go down into the mycelium. But there's even examples where it's upwards of 50%. And that nutrient economy is different between all these different species, different tree species, and time of year. Because in the middle of winter, when the days are really short, the plant's not making a whole lot of carbon, or fixing a whole lot of carbon, because there's not a lot of light. Whereas in the summer, it's probably making a lot more energy and has the capacity to give a lot more to the mycelium. But keep in mind, trees and mycelium are somewhat offset. Mushrooms aren't always produced when the trees are at their peak of making the most carbon. So there's a give and go there that really is not well understood in terms of like what's the pecking order of different mushrooms, how did like mushrooms decide when to form, how do plants determine like what's my preferred mycorrhizal partner? Does that shift over time with like the tree species changing? And it definitely does, because as you see environmental succession and different tree species taking over, you'll see different fungal species becoming predominant within forests. And this is a thing they see all the time with like commercial lumber plantations, where they'll be looking at the types of mushrooms that are occurring with the trees. And once they go from, I think a lot of lumber plantations are seeded with Swillus species, Boletaceae, things that help them establish the seedlings. And then later on, when they start getting more like Amanitas and Chanterelles and Porcini, they know that the stand has matured, and it's then ready for them to like come cut it down, which kills the foragers, because they're like, you just took away my bolete spot. What the hell, man? So anyhow, there's a nutrient economy there, and there's, a vice versa of like what's a sort of a low effort, low return versus like high effort, high return, and then how is that pecking order figured out? So there's lots of competition, and the associations are conditional and not given, and you can screw that up by like fertilizing your soil a lot. You see this all the time in agriculture, where you give a lot of nutrients, and then the plants will kick out their fungal partners, and so that's not necessarily great for soil or microbial diversity. And it has a lot of weird stuff to do with like how we do management strategies too. Like the fact that if you pull leaves away from an environment, like if you just rake your lawn, you may be changing the dynamics of what fungi are in the marketplace with that particular tree, because that tree has its own needs that may be different from the needs of the fungi. And if you're changing the nutrient conditions by fertilizing your lawn or getting rid of leaves or stuff like that, you may be changing what types of fungi are predominantly associated with that tree. So I am going to talk really quickly about sort of this skeptical idea on the CMN, this common mycelial network, and then I'm going to finish up with just a few extra weirdo types of mycelium. I've mentioned a few of them already, but we'll get into that. So there's a great paper put out by Justine Karst and Melody Jones and Hoeksema in 2022 called the Positive Citation Bias and Overinterpreted Results Lead to Misinformation on Common Mycorrhizal Networks in Forests. And this is essentially kind of a scathing takedown in response to this media interpretation that the Wood Wide Web is all of these trees like hugging and sharing underground, and that everything is like hunky-dory, and that like old trees help young trees, and everybody's like trading stuff back and forth. And it's based on sort of three main assumptions and claims that this paper takes a very skeptical eye at. And one is that common mycelial networks are widespread in forests. Second one is resources are transferred across the common mycelial network, especially with regards to like seedling establishment. And then mature trees are preferentially sending resources and defense molecules to their offspring. And all three of those things have been demonstrated to some degree or another, but if you look really critically at what's actually been done, there's still a lot of outstanding questions. And so these scientists are absolute legends in their field, and I think they took it upon themselves to try to add a level of necessary nuance to the conversation, such that things in the media have basically been saying that like mycorrhizae are the most incredible things, and like, mycelium is the internet of nature, which again are good gateway concepts, but is not necessarily illustrative or accurate when it comes to describing what's happening down in the soil. So I'm going to just kind of quickly run through some of the points they made in that article. It's a really good one. If you have a chance to read it, you should. It's pretty academic, and it's focused more on like researchers, and I think it's really meant to help people who are in the field understand how they could do research better. But I really wish that some journalists would read it too, because I still see this constant movement of articles that are saying that like mycorrhizae is all helping stuff hug underground. And you're like, well, do you understand like arbuscular mycorrhizae versus ectomycorrhizal? And that's part of the reason I spent so much time talking about the difference there between AMF and ECM fungi, and like the habitats they inhabit and what they're particularly good at doing for the host plants. So number one,
it's really hard to measure and evaluate mycorrhizal networks in nature. Like I talked about, like Merlin Sheldrake spent a lot of his PhD crawling around on the forest floor, and he was studying a microheterotroph, and I don't know how well he ever really managed to like map the networks of stuff, but it's incredibly laborious to try to crawl around on the forest floor and dig up like microscopic threads. How do you really track that? It's experimentally challenging and technically very difficult to do. To date, and this is what was quoted in this article, is there's only like 2 tree types out of something 70,000 plus, and only 3 ECM species that have been mapped. So we don't really know how far the common mycorrhizal network goes within a forest. And there's big questions about like, what is that in a boreal forest versus a temperate forest versus like a mixed hardwood conifer forest versus in a grassland? Can we really say that a whole grassland is connected by the mycelium? Maybe, but maybe not. Can we say that, a forest that has lots of different tree types is all still sharing nutrients when those trees are in clear competition with one another, and then each tree has like 20 plus species of fungi associated, some of which might be arbuscular, some of which might be ectomycorrhizal? Again, we don't know, so we can't really say that the common mycelial network spans out in all directions and connects all these trees. There's a thing from the Fantastic Fungi movie where there's this great graphic of them spinning around underneath a tree, and there's all these sort of lit up branches of this mycelial network. And like, that was inaccurate because most plant roots are fairly high up in the soil. They don't go super deep for the most part. And then, yeah, are they really connected? We don't know. And that's kind of the point that they're making, is we don't have enough evidence to say that mycorrhizal networks are widespread and interconnected. There's a chance that they do, but there's also a good chance that maybe instead of being interconnected, they're competing. So the reality there is a lot more complex than the idea that, it's this big contiguous network. Doesn't mean it's not true, and there might be instances where it is true, but there might be other instances where it's not. And so again, necessary to recognize the complexity and the nuance of the whole common mycelial network concept, right? The second point was that about nutrient transfer, and it was talking about the movement of nutrients down in the soil. And a lot of the experiments that have been done demonstrated some level of nutrient transfer, but if you take away the mycorrhizal relationships, you can still see nutrient transfer in soil. It happens on its own. In theory, if the mycorrhizae is there, it is facilitated and the nutrient transfer happens faster. But in a lot of these experiments, they were done either in little pots in labs or in nature, but the seedlings were wrapped in like mesh bags, and there's a lot of what they call confounding factors. Controls weren't necessarily done properly, or there's things that kind of complicate the results in terms of understanding, is nutrient like transfer really happening? Is it happening by the CMN, or is it just happening through the soil? That's still sort of an open question within the research, and there needs to be better controls and experimental setup to like understand this, and that was one of their main points. And it's not really clear of like the extent of AMF versus EMF networks, how long they extend, do they interact, how much nutrients can you transfer from EMF versus AMF, or just between different species, right? We don't know. The efficiency of those transfers, and then also like I was talking about the time of year, the season, the marketplace economy there for mycorrhizal relationships is definitely going to affect whether or not nutrients are getting transferred and how they're getting transferred and how far they're getting transferred. So again, we lack the necessary data to know, and that was one of their main points. And then their third thing was like the mature trees preferentially helping out younger trees. And so there's a little bit of data out there about some arbuscular mycorrhizal, I think it was tomatoes and beans. They showed like one plant got attacked, and then through the mycorrhizae was able to like transfer a chemical defense signal to the other plant that the pest was around, so it was able to start producing molecules that would deter predation by a pest. But that was done with like 2 tomato plants in pots. And I think there's also been some follow-up studies where they said that like, oh, you didn't actually need the mycelium present. The plant produced the molecule, and then the molecule just like passed through the soil to this other thing. Sometimes it happens with aromatic volatile molecules as well. So plants have a lot of different ways to communicate with each other that may not be fully dependent on fungi. Do the fungi facilitate it? Maybe. but we don't necessarily have the data to prove that because of these confounding factors and lack of good controls in the experimental design within the scientific community for the way that people have been studying this. And so there's, what they said in this was that, to date, there's no good evidence that mature trees are sending any kind of signals to seedlings via the common mycelial network. So that idea was something that they pushed away from. And they said sort of within the literature, and this is across like 1,500 publications, there is a pattern of overstating results, unsupported statements that had a positive bias towards, assuming this anthropomorphic assumption that the mycelial network was helping all of the trees of the forest as a whole. And there's a lot of sort of what they called confounding factors of things that weren't totally clear. And so it's a good skeptical step back from this concept of the wood wide web and the idea that, everything is in this like big mutualistic symbiosis in the forest. And it doesn't mean that that's maybe not true to some degree, especially within like big conifer forests where a lot of the trees are the same species. A lot of them, there's mixed ages, and you certainly see that. Like if a tree dies, sometimes the stump will resprout, or like something gets diseased and then it'll eventually come back. And you're like, well, how did it come back? And like, well, they found that some nutrients can go into those trees from other trees. But the question is it just osmosis or is it intentional? And that's what we still don't fully understand. Is it just like a concentration gradient thing, or is it like, ooh, hey, that thing needs to go over there? And two, within this common mycelial network, there's idea of like different trees and different things acting as nodes. Plants like microheterotrophic plants that are preying on the mycelium of fungi may also be helping to facilitate the transfer of nutrients or even just extend the mycelial network. And that's a question that's still outstanding because, again, it's really hard to study this kind of stuff and we don't fully understand it. So I think there's a definitive possibility that, trees are facilitating communication, that they can help transfer nutrients, and that, there may be some preferential stuff where if you have an offspring of yourself that maybe you're going to try to help it. But I don't think that we can say that conclusively, and there needs to be a lot more experimentation done before we can make those statements with any level of certainty. So again, this is the scientist in me sort of saying, well, but, got to show it. It's always good to have data. Okay, so I'm going to finish up the two main types of the two main types of mycorrhizal relationships are ectomycorrhizal. It's like 2% of plants. There is also arbuscular mycorrhizae. That's like 70% of plants. There is ericoid mycorrhizae, and that's associates ascos and basidios, mostly associated with ericaceae, blueberries, cranberries, bog stuff. It's poor soils, cool, wet environments, and lots of saprotrophy. So those are important commercially for, berry farmers. There's orchid mycorrhizae, and that's mostly basidios that are epiphytic. So again, I don't totally understand how mycorrhizal fungi is like up in the treetops where orchids live, but there's tremendous amounts of orchid diversity. So that's why it's like 10% of plants have orchid mycorrhizal associationships. And usually what happens there is an orchid seed doesn't have a lot of nutrients in it. So most plant seeds are full of starch and protein, and that's why we eat grains, because we get energy from them. You would not get very far from eating orchid seeds because there's nothing in them. Instead, orchid seeds germinate and start sending out signals that sort of tricks this mycorrhizal fungi to come and try to like do some saprotrophy on it. And then the orchid seed hijacks the mycelium and fills itself up with all the nutrients and carbon and stuff it needs to germinate and form a plant. And so some orchids only do this when they're young. Once they've established themselves, they just do photosynthesis and they don't need a mycorrhizal partner. But there are some examples of orchids that continue this mycorrhizal relationship and may even give some benefit back to the fungal host later in life when they're starting to do photosynthesis. So it's interesting how there's some examples of mutual exchange and some where there aren't. And again, there's probably a lot of different evolved versions of this orchid mycorrhizal relationship that have happened over last several 100 million years. There's arbutoid. Ericoid, by the way, those are endo. They form sort of arbuscular-like structures within the cell. Orchid mycorrhizae, I believe, are also inside of the cell. Arbutoid mycorrhizae is formed by basidios, and that does form a Hartig net around the sheath around the plant, but then it also penetrates the plant cells. There are monotropid mycorrhizal relationships, and those are the ones that microheterotrophs have with the fungal partner. And in that sense, it does not penetrate into the host cell, but the microheterotrophs form little like pegs, what is sort of nutrient exchange things, and those increase when the microheterotroph is going to flower. So it asks for more nutrients basically when it's going to form a flower, because the rest of the time microheterotrophs are little like rhizomes down in the soil, not doing much. They don't do any photosynthesis, so they're entirely dependent on the carbon they get from the mycelial network. And then there's Ectendomycorrhiza, which is formed by ascos and basidios, and it's sort of an intermediate form between AMF and ECM. It's more common in conifers, but can also occur in angiosperms, and it's sort of a hybrid because it's similar that it forms like a Hardig net, but then sometimes it also penetrates. And from what I've read, it's more of a descriptive term than like a really well-defined category. So those are some of the other types of mycorrhizal relationships that are out there. And again, it's probably evolved many times over the last 400 million years. And so there's a lot of diversity of mycorrhizal relationships. And we understand so little about it because it is down in the soil. It's hard to study. And, you know, everything I've told you is a tremendous amount of information. And there's way more out there in literature than I could even really try to accurately summarize in a podcast. But there's my take on it. I think it's fascinating. I've tried my best to talk about some of the nuance there and describe the challenges that are ongoing in the field, do some literature citation, and, you know, give credit to people who've done this research. But there's still so much more to learn. And also, just importantly, mycorrhizal relationships are a keystone for all plant life, pretty much. And the only plants that aren't keystone for are these legumes, and even legumes will sometimes benefit from mycorrhizae they don't necessarily associate with their roots, but it's just nearby anyhow. So that's another thing. There's some percentage of mycorrhizal relationships that aren't on a host plant, but are around and still benefiting a plant potentially by the fact that they're nearby, and they're creating good conditions within the soil by cultivating good bacteria and mineralizing the soil and holding lots of water and stuff like that. So mycorrhizae are absolutely crucial to pretty much all plants on Earth, and thus life on Earth. So we have fungi to thank for that. And I don't think that's an overstatement by any means. So thank you for joining me. Again, thanks to North Spore for sponsoring this episode, and appreciate you guys being here. If you enjoyed this podcast, please remember to give me a positive rating on whatever platform you're watching on. Share with your family and friends. And yeah, if you want to support me, you can always join my Patreon. You can follow me on any of my social medias, Instagram, TikTok, YouTube, Facebook, Pinterest, all of the things. I've got merch on my website, so that's great. If you want to order anything from North Spore, you can use my discount code FASTENBYFUNGI in all caps, and it'll save you 10% on your order. And again, big fan of their immunity blend and some of the cold tolerant mushroom varieties that they've got here in winter. The snow oyster, the pioppino, king trumpet are all really good ones to grow this time of year. So I bid you guys adieu, and till next time, I'll see you soon. Okay, bye guys. Cheers.